So today we're going to talk about the tools for doing this brain mapping that are emerging. Okay, and I still weave in a little bit of history there, but this is going to be more about physics and engineering today. Okay, so people that I'm working with, 6 different groups, and there's more, but these are my close collaborators. Let me just very, very quickly, not spend too long, let's spend 5 minutes just reviewing what I said about functional connectomics. If you stain 1 in 1,000 cells, you can make pretty pictures like Ramon y Cajal did on the right. Today we can stain all the cells, and it becomes it's apparent how daunting the task is of actually making sense of this incredible tangle. fMRI, cognitive neuroscience, magnetoencephalography, EEG, these things all do ensemble averages of regional processors. The tools haven't existed yet for actually understanding how the bits are flying in these regional processors in the brain. So we're going to talk about that today. From a physical science standpoint, the brain is a whole bunch of fields, and so it's a very, very complex measurement problem. Signals are very weak, but that's the way we can think of it. And I talked about different technologies for mapping the brain. fMRI, where individual voxel, a 3-dimensional pixel, averages over 100,000 neurons. Intracellular recording, where we can study in great detail individual neurons, but we can't really scale up very well. It's really hard. Extracellular recording, where we can actually scale up to a lot, and I'll show you what we're doing there, but the penalty we pay is the signals are weak because we're in the extracellular environment. And even Nobel Prizes are obtained for measurements on individual neurons, but it would be really nice to actually have tools that have multiplicity, allowing us to record them more than 200 neurons at a time to understand what's going on in brain circuits. Optical functional imaging looks good. You can measure up to 30,000 neurons, but it suffers from the fact that the brain is a very opaque and scattering tissue, so you can't go very deep. Several hundred neurons are obtained by doing this random access scanning, and thousands of neurons can be obtained by this light sheet microscopy, but again, this only works in transparent tissue. So for optics, basically, most of the brain is opaque. So I'm going to talk about how to break that paradigm with nano-enabled tools. First, we'll talk about electrophysiology. Okay, so how was that for a review? That was like less than 5 minutes, right? I could have done my talk in less than 5 minutes. Nobody would understand what I was talking about. So one of the things that we got hassled about when we said we wanted to map the activity of the brain was, how big, how extensive could our measurements be? And so we stepped back a little bit and we said, okay, we want to find some regional circuits in the brain that we can actually study. And this still is sort of an open question because you've got this pulpy mass. How do you actually say that one part of the brain is a regional processor? So the question, what constitutes a brain circuit? So I'm going to give you examples from current research of 2 different types of brain circuits. First is distributed functionality across brain regions. Somehow there's some sort of non-local computation that are going on in different parts of the brain, but that it's coherent. And then the other idea is a regional processor where, in fact, all the synaptic connections are very dense in some small region and then sparser outside. And so I'll give you that example. So first, let's talk about distributed functionality across brain regions. So associated with sleep is the hippocampus. This is a cartoon of the hippocampus of the rat. This is from my close collaborator Nanoscience group at Caltech. And remember that layer of cells that I showed in the rainbow picture? That's called CA1, and that's at the top of the hippocampus. It's some millimeters away, 3 millimeters away or so from the cortex, the outer surface of the brain in the rat. What my colleague did several years ago is he used these twisted wires, and he put very carefully something like 28 of these twisted tetrodes into the brain. And that allowed him to sample across a region. Not densely, all the neurons, but a sampling over some spatial locale in the hippocampus. And what it allowed him to do was, for the first time, study these oscillations, these synchronizing oscillations that occur in the hippocampus, called theta waves. And he found out that everyone thought that these theta waves were synchronized throughout the whole brain. But in fact, because he topographically sampled, he could actually see that these theta waves were traveling waves in the brain. And so people had studied these for like 50 years, but with one electrode. So here's an example where if you have distributed electrodes, you can begin to see that there's some sort of emergent pattern, some emergent coherence that's happening. Yes, please. Theta waves are slow oscillations. They're on the scale of milliseconds, longer, they can even be seconds, in the brain where lots of neurons are involved and they're spiking. And it was assumed that they were all spiking coherently, but now it's known from their work that they're actually traveling waves of coherence. And these theta waves are thought to be involved in the storage of memory, and they synchronize the firing of local neurons. Okay? Yes?
So currently there are like two different hypotheses about these theta waves. They either think like, this background theta wave like oscillation is sort of necessary for the function of the brain. But there's also like this epiphenomenon theory that like it's just random and it's just there and it doesn't actually serve any purpose. Do you have any?
Usually when people say that things that are very well defined and coherent in biology are just random and junk, they're proven wrong. Like we thought that whole parts of the genome were just trash. And now it turns out this is very essential stuff. So I believe that there are theories, but more work needs to be done with extended data sets. But I believe that these things are very important to memory formation, for sleep, and so on. That they actually allow a global sort of setting of the activity of the brain. But I'm not an expert in this area, so this is just a guess. But we'll get more data and we'll find out. Thanks for the question. So a second example of a brain circuit is within a region. And the example comes from sort of the formation during embryogenesis. Basically, after fertilization by the sperm of the egg, the cells divide, and from progenitor cells, more and more cells, daughter cells, are generated. And these daughter cells then form structures. And in the brain, one of the structures they form in the cortex are things called cortical columns. And this is a cartoon from Andreas Tolias's group of these cortical columns in the cortex and how they are thought maybe to actually form some sort of structure, ontological structure that actually happens during development. Okay? It turns out that these cortical columns have many more connections, many more synaptic connections within the cortical column than outside. So it seems like, intuitively, this sounds like all this extra wiring, connecting this region to itself seems to implicate it as some sort of like regional processor. Here is a picture of the cortex, and I put up that great quote from Ramon y Cajal once he actually started seeing things. This is colorized, a diagram, and there are 6 layers in the cortex of the higher primates, including us. This is the outer part of the brain. Many different cell types are distributed and connect to each other in the cortex. Okay, so these are the kinds of different daughter cells that can be generated in cortical columns. Remember I talked about these multi-patch experiments? So the experiments that Xiaolong Zhang was doing in Andreas Tolias's group he was looking at the connection strengths as he moved away from a neuron that was in one cortical column, or assumed to be a cortical column, further and further away, and measuring the connection strength by multi-patching. And through a whole series of laborious measurements, he notes that sort of with a diameter, or sorry, a radius of about a half a millimeter, in fact, these connections actually decay. So this seems to be further evidence that there's some sort of local connectivity that's very strong. But once you move a distance of about a millimeter away, that connectivity dies off. There is a number that I want you to remember for this talk. If you have a cubic millimeter of brain tissue in the cortex, in the mouse, that's about 100,000 neurons. And so that's going to be the target for the second part of the technology that I talk about, to record from 100,000 neurons in a cortical column. Okay? All right, so now we get to the nanotechnology. There are two research fronts that we're following. We're trying to massively upscale extracellular electrophysiology, and we have a new paradigm called integrated neurophotonics that will take us even further. Okay, so let's talk about extracellular recording. I mentioned that the state of the art as of about 3 or 4 years ago was something like about 64 full-time channels that we could record from 64 neurons, or maybe more if we triangulate, as I'll talk about in a moment. This uses microfabrication technology to make these kinds of probes. But still, you know, that's a small number compared to the hundreds of billions of neurons that are in human brains. And so we've pushed further, introducing nanoelectrical systems into our probes. This is a probe, small cross-sectional diameter, that can be tolerated by the brain. If you insert it very slowly over the period of days, the brain tissue can move away. You won't actually cause bleeding or scarring or immune response, and you can record for long periods of times from these electrodes. And here's the electrodes. This is coated with a plastic, basically, around, so only the electrodes are exposed. Now, the problem is that, or one of the issues is that we have to make these electrodes a specific size, about 100 microns squared in area, so let's say 10 by 10, if you made them square. And you don't want to go smaller than that, because then the impedance gets really high of those electrodes, and the signal-to-noise ratio for recording spiking degrades significantly. So we've got to stay around 10 microns. So that means a good fraction of the probe diameter is going to be, the cross-section of the probe is going to be these electrodes. But the only way we can get hundreds of connections up is to actually use nanotechnology to make very fine wires up the shanks. And so these are some of our first efforts using 300 nanometer, a third of a micron electrical interconnects, running a half a centimeter up to the head of our probes. And these are the guys that did it. Gustavo is just about to finish his PhD this summer, a superstar in the lab. Eugene Lubinov, who is a longtime researcher, he's the one that discovered with Thanos those traveling theta waves. He's a senior scientist in our combined groups. And Derek Chi is a research engineer. So Gustavo and Eugene and Derek have basically made 256 channel layers and stacked these layers to make the first full-time 1,000-channel probes. And this is over the last couple of years, culminating now. They're actually in the laboratory using these probes to actually make recordings. Now, the nanoprobes is not the only thing that you need. You need a system. And this is sometimes is lost on people that get very enamored about nanotechnology and nano devices, that a nano device is only part of a whole system that's needed to actually interface. The second thing you need is you need boxes of these things. You can't have a hero device that a student works on, you know, for their graduate degree or something and makes 3 of them or something like that, writes a paper telling about the technology, but then when he takes them and tries to implant them, they get fouled and experiment over. No more probes. Now you need boxes of these probes. So we'll talk about that. Part of the system is actually the electrical recording system that you connect to, and the thousands of connections that have to be made to the probes themselves. And so I won't go into the details of the technology. There's many, many fine details that you have to worry about, engineering details that you have to worry about in order to make these systems. The chips themselves push the state of the art of low power, very large scale integrated CMOS electronics, and Ken Shepard actually my first graduate student a number of years ago, now a chaired professor in electrical and biomedical engineering at Columbia, is a world expert. And actually, he worked at IBM for many years designing computers after he got his PhD in physics with me. And now he is using all this expertise to actually do sort of off-axis things, not information science, not computation science, but actually using this technology to do stuff in biology, which is really fun. The other part of the story is if you want boxes of chips, if you want boxes of these micro and nano probes, you have to partner with a foundry, a plant that actually builds the chips. It turns out today the cost of a plant to build computer chips, the entry fee is about $4 billion. That's what it takes to actually have a plant like this making the chips. Most people, if you ask them, say, I'm a wacky physicist, I want to make some nano devices using your tools. Click, bzzz, you know, oh, that's what it sounds like in the US when people hang up on you. So I found, actually I was invited to this research foundry in France back in 2006, and we started this thing called the Alliance for Nanosystems Very Large Scale Integration. And the first thing we did was make a standard process for making nanomechanical devices, something I had been working on for about 15 years in my group. And after a year and a half, we had our first standard NIMS process, and one wafer with something like about 6 million devices, just one wafer, exceeded by several orders of magnitude the total integrated number of devices that had been made everywhere in the world for the previous 15 years. So that's what very large scale integration can do. So over the past couple of years, we've used this technology now to take those prototype probes that Gustavo and Derek made in the Kavli Nanoscience Institute at Caltech, and now to make them robustly using foundry processes. And so this is the first, this is one of the wafers in our first batch of 200 millimeter or 8-inch wafers that has thousands of these nanoprobes. And you can see how pretty it is when you actually use this perfected technology for making micro and nano systems for neurotechnology. So where are we in terms of pushing towards massively multiplexing electrophysiology? Today we're at a 1,000-channel system, which exceeds the state of the art by a factor of 4 or 5 from that plot that I showed you that up till 2014 or 2015 was only a couple hundred. We have a proposal in that we hope is going to be funded that with Ken and my group and the Kavli Nanoscience Institute and our collaborators at CEA Leti and Grenoble, that foundry that I talked about, we think we can get up to 10,000 channels with custom electronics chips for doing the electrical readout and also custom nanoprobes. And with more funding, in about 5 years, we believe it's possible to go up to about 100,000 channels. What about 100 billion? We're still a million off from that. Well, I don't think electrical recording can get there. And the reason is the following. It's not any sort of fundamental physics or fundamental, principles of engineering. It's basically practical issues about how small, using the state-of-the-art of computer chip technology, how small you can make the wires and the probes and everything else, and how many of these probes you can actually put in the brain and how well the brain can tolerate it. The rule of thumb that empirically people seem to have found is as long as you don't exclude more than, or as long as you exclude less than about 1% of the brain tissue when you do these implantations and you do it slowly, the brain can function okay. But if you go more than 1%, then you're starting to really perturb, destroy, cause dysfunction in the brain with this bed of nails that you're putting in. Okay? Yes, please.
What brains are you testing this on?
So mice, rats, and then with great pause and forethought, primates, non-human primates, monkeys. So Andreas, I think I showed a trace of him, some of the work that he's done in macaques. Also insects, turtles, we have there are interesting comparisons evolutionary. For example, the turtle has only a 3-layer cortex, and it's kind of interesting to understand how that's organized compared to a 6-layer cortex. And so we have a broader circle of collaborators that are actually studying other things. But primarily, everything is initially studied in mice, then translated to rats that are freely moving. That's harder. And then once we actually perfected in that, then we think about actually doing things in macaques. Yes?
Do you think because of how different each individual brain is or appears to be, that even if you found out kind of the workings of one brain, then that could be completely different?
I don't think so. I mean, I think the structure, the wiring, is probably completely, or, you know, there can be huge variations. But we have similar behavior. You know, we have similar gestational phases. You know, when we are born, we grow up, we learn language, we learn how to function, we learn how to socialize. There are many commonalities that we have. And those must be reflected in some sort of computational motifs in the brain. That's what we hope. But yet, we have not yet figured how to extract this sort of leading order behavior, you know, that we see that's common, and reflect it or understand what's common in terms of, you know, what's conserved, what's common across all of us in how our brains compute. And when we have more data, we'll be able to look for these kinds of correlation between people or between mice to start with, and then ultimately between different species, we'll see the differences, and then perhaps between people. Similar sort of thing happened with the Human Genome Project, looking at the commonality of the genome, looking at differences, right? Small differences that emerge turn out to be all important.
So far you've been testing on like rats and mice.
Yeah, and insects.
To date, how has with those brains, how have those brains tolerated the
It depends on, this is a good question, and it's essential understanding this if you're saying you're going to use implanted technology to do your experiments. So the rule of thumb is this, and our sort of overarching charter for what we actually engineer, those tetrodes, which are about 35 microns in diameter if you use like 10 micron wires, if they're implanted very slowly, you know, over the period of weeks, you know, just sort of microns per minute or something like that, you know, down to the multiple millimeters to get to, for example, the hippocampus, you can do long-term recording over months, maybe even years. If you put it in really quickly and you cause bleeding immediately will trigger an immune response. The brain, the tissue will actually recognize the probe as a foreign material, not self, but, you know, outsider. It will create scar tissue to basically encapsulate it, wall it off from the rest of the body, and your signal-to-noise ratio goes to 0, right? Okay. Yes?
Does the layer of the cortex have anything to do with how controlled is the animal?
Well, so I'm not an expert in this, but yes, I mean, the higher mammals actually have this 6-layer cortex, whereas reptiles, you know, have 3. You know, so I forgot the name for the. Anyway, I'll remember the nomenclature. This is where the jet lag comes in. I can't remember basic things. Yes. Yeah. No, they haven't been. That's a great question. They have not been. Yes.
Are these probes single use?
No. Well, yes, they are single use. I'm sorry. I said no. I don't know why I said no. Yes, they're single use. Absolutely, they're single use, especially in, you know, valuable species, like if you've studied the behavior of a particular rat and you're keeping it alive, you know, for a couple of years. You don't want to insert a probe a second time. These things actually have to go in a very aggressive chamber to actually sterilize them before implantation, after the surgery, to open the skull so that the probes can go in. So this is done. My colleague Andreas Tolias and Thanos Siapas both, it's interesting, they were like both mathematicians before they became neuroscientists, and now they're expert surgeons, you know, for animals, right? I just can't get near that stuff. But anyway, they have a whole series of, you know, highly sterilized protocols. They have a surgery room for doing all this stuff. No, you don't want to stick some old probe that's been in somebody else's brain in your brain, or your favorite rat's brain.
Is human testing ever going to happen, do you think?
Well, so I mentioned yesterday that human testing is happening. And it happens, for example, I'm working in Grenoble as part of this CEA, which is the French Department of Energy. They have a thing called Clinitech, where the Department of Neuroscience at the University Hospital in Grenoble, a group of people, in fact, the first inventor of deep brain stimulation for Parkinson's disease, they created a little mini hospital that has 3 beds called Clinitech for actually translating it. This word translational means taking into medical practice new technology. So translational medicine, translational neuroscience. And so they have some new technology for actually imaging, locating where the brain tumors are. We're actually involved in developing some of that technology. And if the patient, because you have to open the skull to actually find the brain tumor to excise it, if the patient gives permission for there to be some research done locally where the tumor is, then other things can be explored. But again, it's only sort of in almost hopeless cases where this is actually done. It's not in, you or me or healthy people. Pardon me?
Mass scale human testing, is that ever
Mass testing, yes. Probably, I don't know. I'm not sure in our lifetimes or not. Yeah. So there are lots of people that have auditory implants, for in the cochlea, retinal implants, and those all work if you're cabling to your brain and to the visual auditory cortex are actually functional. But what happens if those are damaged? And that's the reason why you're blind or deaf. Well, then you would like to actually implant in the auditory or the visual cortex to actually stimulate directly there. Pattern stimulation to create stimuli that's similar, if not identical, ultimately to the stimuli that comes from the retina, for example. So I think that's the future. It's going to be a long process of getting medical regulations. We're not going to want to test out flaky stuff like we're doing today, on people who otherwise would be okay. Yes? It could be, yeah. I think the final answers or complete answers for these things are not known. We're building some, so, I mentioned this friend or foe sort of response that the body has, the immunological response to, foreign objects, antigens, or a probe, right, when you stick in. So we're actually making some probes that can look for things called cytokines, which are released by immune cells or as part of the immune response of the brain. And what we want to do is with these chemical-based probes to actually correlate the morphology of the probes, the insertion, all the things involved in actually implantation, and try to find if there's a sweet spot in parameter space. There's a correct protocol for actually implanting these without eliciting an immune response.
To date, has there been any differences between species when you insert them, the tolerance of the brain?
I don't know the answer to that question. I'll have to ask my neuroscience colleagues. I've mostly been working with the probes that we've been building on rodents, and we know in those cases one protocol that allows us to do chronic recording. Chronic means long-term recording. I'm sorry, can you speak up? Project. Yes, absolutely. This is critical. Stimulation and recording are two things that are absolutely essential to do. I mean, the first phase is to observe. And if you observe and you can't actually perturb, then you're sort of watching the natural response of the brain. You can actually create cognitive tasks or tickle the whiskers of the rat and see various neurons firing, give visual stimuli. But What's really exciting and interesting is to do closed loop control, to actually then go in and excite neurons directly and see if you can actually elicit responses and record those responses. And so stimulation and recording are absolutely two things that we're targeting. I've only been talking about recording, but stimulation is in there as well. And the chips that Ken Shepard and his group are building not only allow us to record, they allow us to stimulate, and they allow us to do amperometric sensing of local chemicals in the brain that are released. It's not just electrical activity, but the brain, remember, it's brain fields. There's all sorts of things that are going on in different domains in the brain simultaneously. And to get a complete picture of how the brain computes, we've got to look in all the domains in which the brain operates and see this correlated behavior. May I go on? No, I love your questions, so keep asking them. Okay, so why do I say we can't probably practically get beyond 100,000 neurons? And we have to look in a little bit of detail at how we actually record from many neurons. It turns out that there's a technique that is akin to triangulation when you have multisite electrodes that you can use to separate signals from adjacent neurons. So this is a cartoon of a tetrode inserted. These are supposed to be neurons. Pyramidal cells is the reason why they have that funny shape in neural tissue. And these neurons are going to spike. We have different channels. And we're going to actually see from these different channels, because these wires are slightly different positions, we'll see different signals. They're not going to be exactly the same. But we can actually use waveform recognition that this particular neuron will generate something that looks slightly different at each of these electrodes, but we can correlate that. And we can then also do triangulation, and this will allow us to separate amidst this cacophony of all sorts of neurons firing. We can actually localize, at least in some parameter space. We may not exactly know spatially where it is, but electrically, in signal space, we can actually recognize the firing of a particular neuron. We call it a unit, not a neuron, because it's sort of like in a configuration space. We can separate out the signals that are coming from one particular neuron that's somewhere. So this spike sorting technique and these multisite electrodes allow us to yield sorted time records of the firing of individual neurons. So we can ask the question, over what length scale is this protocol effective? And well, a bunch is actually known about this from sort of single and few neuron recordings. This is, all the wonderful background. Even though I sound like I was critical, we haven't recorded from many neurons. We know a lot about how neurons work. And this is important. It turns out in a volume or a diameter of about 50 microns around these electrodes, we can resolve with good enough signal-to-noise ratio individual units. We can do the spike sorting. But beyond that, essentially, because the signals decay in the ionic solution, remember I said you start from tens of millivolts, and then by the time you get 50 microns away, you're at tens of microvolts. And pretty soon, the signal is so small, the noise of your amplifiers and everything swamps it out, as well as all the background noise, the cacophony of all the other neurons firing. And so the rule of thumb is that this spike sorting works. Sometimes you're lucky and you can separate from one electrode or one tetrode, you can actually see 6 neurons independently. But most of the time, you get something, on average of about 1 or 1.5 units per electrode. So I mentioned these cortical columns, 100,000 neurons. That's in 1 cubic millimeter. If we record electrically from them, 1 cubic millimeter, and we only get a multiplicity of 1 to 1.5, we're going to have to put something like 75,000 wires into that millimeter cube. That's why I'm saying that you can't do it. Okay? You can't do it and yet still preserve this rule of thumb that we don't want to displace more than 1% of that tissue. So we need a different paradigm. How can we record locally from more than 100,000 channels? How can we record from all the neurons in something like a cortical column? Can we access other brain fields besides the electrical domain? And, super important, we need to make devices that aren't just hero devices that get students a PhD and some nice paper. We actually want to make technology that can be mass-produced and fuel a revolution in how experimental neuroscience is done. And that's where this idea of integrated neurophotonics comes in. So I mentioned that free space optics won't penetrate the brain well. In fact, it doesn't more than something like a millimeter. And I mentioned last time the reason for this arises from both absorption and scattering in brain tissue, which is wavelength dependent. So our paradigm is the following. Let's break this impasse by implanting pixels, all of the elements of an imaging system. I'm going to call these emitter pixels and detector pixels. I'm going to use the same form factor of these ultra-narrow shanks that we can implant in the brain, but replacing all of the micro and nano electronic components with integrated nanophotonic elements. So here's a cartoon of what we're building, and I'll show you some examples. This is work in progress. This is, again, the CA1 and the hippocampus of the mouse. Here are some probes that are about 3 millimeters long, and again, tens of microns, a few tens, maybe 10 or 20 microns in width, so as not to create this immune response upon the implantation. If you zoom in on what we're trying to do in this cartoon, this really is a rainbow picture that I overlaid with the right scale factor for our probes. Here are the soma, and you can actually see the processes, the axons and the dendrites. What we see here on this probe is illumination emerging, and these green things are photodetectors. In particular, we have photonic waveguides that guide light in these nanophotonic elements on these probes down to particular places where it emits. And then the green things are single photon avalanche diodes. They're photon counting detectors that then send electrical signals back out every time they capture a photon. Please.
What material is used to construct those detectors?
Silicon. So lots of crazy, new, and exciting materials are used in nanoscience. But if you want to make something that's going to be mass-produced, stick with silicon. I didn't know that a few years ago. Well, more than a few years ago. But when I started deciding that I wanted to mass-produce stuff, I realized trillions of dollars, maybe more, have been spent on silicon electronics, silicon-based electronics, and now silicon-based photonics for telecommunication. So if you want to mass produce and you don't want to reinvent the wheel, it took 50 to 60 years to actually create this mass production technology where, it's microcents per transistor or less, femtocents or something, I don't know what it is. But we're not going to have that with other materials in our lifetime, right? So let's stick with silicon if we want to mass produce stuff. So the concept of photonic probes, it's again this sort of bed of nails in at the micro scale of the form factor. It's a 25 shank assembly, 3 millimeters to get down to the hippocampus in the mouse, active regions with these E pixels and D pixels over the last 2 millimeters. And what we have been funded to do by the Brain Initiative, I'm happy to say, is to create a system that has 25 shanks and a total of about 2,000 detectors and 2,000 emitters. Not 75,000, 2,000. With a multiplicity of 50 to 1, we can record from, if we can get 50 to 1, we can record from all 100,000 neurons in a cortical column. This whole thing is a millimeter on a side, and so will displace less than, or of order 1% of the brain tissue. This shows a picture, and this shows an intemperate promise that I inadvertently made to get funded in our proposal. Be careful what you promise because you may be held to it. I said that we were going to make 10 of these as modules and tile them together to record from 1 million neurons in 3 years. And now I have to live by those words. Okay. I think everybody would be happy if we recorded from something approaching 100,000 neurons. But unfortunately, there's inflation in the promissory department of people doing science these days. It's just really hard to get funded. There's a problem, though. I talked about brain fields. Does anybody see photons in this picture here? No. The brain doesn't work in the optical domain. So how do we actually interrogate the brain optically? Well, we need transduction. Action potentials for the brain we can record by sticking wires in the brain. They're in the electrical domain. To translate them into the optical domain, we need a reporter, either a molecule that's induced by the protein-making machinery of the cell, by genetic transfection. You hijack the cell and make it make some molecules that are going to actually report on the activity. Or you can use nanoparticles, functional nanoparticles. So what do these things do? These things basically, in the presence of either a voltage or some chemical, change their optical properties. And once those optical properties change, those optical properties are then going to be modulated by the brain activity. Now we can interrogate this with light and see how those optical properties change. And the change in optical properties is our proxy of telling us the brain activity in various domains. Electrical, chemical, force are all possible. So for our paradigm of recording using these integrated nanophotonic probes, we're not, right now the reporters that are available to us are really quite limited. But there's one that's actually been used for all the functional imaging that I've talked about, the raster scan 2 photon and the light sheet microscopy. People are using calcium reporters. It turns out that associated with the spiking of the neuron, ion channels open up to reestablish the potential, the resting potential of the cell, and this gives rise to a big transient of calcium concentration in the cell. So associated with each action potential, each spiking of the neuron is a temporal spike in the calcium concentration. And so people have figured out how to make essentially calcium binding elements. And when the calcium binds, it stresses or changes the properties of chromophore, something that lights up when you shine light on it. And so you can interrogate the calcium concentration in time by actually using optics. This is maybe too much details here, but basically these particular chromophores called GCAMP6 that we're using, we stimulate them in the blue and then they emit, they fluoresce in the green. Okay? Great question about stimulation earlier. That was recording. How do we stimulate in the optical domain? It turns out that there's ion channels. This is meant to be a picture of the cell membrane, and these are ion channels. It turns out that there, I think it's like some form of sea urchin or something, has light-sensitive ion channels. And if you figure out the gene for that, you can actually transfect cells, and all of a sudden the cell starts making ion channels that go into, are transported and implanted in the cell membrane, and now that cell can be hit with light, and you can cause it to fire, just as if you had given it a little electrical impulse, but now it's in the optical domain. So I call these things optical effectors, and therefore creating local stimulation of neurons. This field of optogenetics that you may have heard about uses these genetic constructs, to actually create either recording or stimulation using optical signals. And most of what's been done is just using optical fibers implanted in the brain that give a big blast of light. And this, what I call a bolus of light, a big dose of light, creates an abnormal excitation of a whole bunch of neurons, completely unnatural except for things like epileptic seizures or something like that. You never see global activity like that. So we need to do something with more finesse than actually exciting with a bolus of light. And that's where these nano-integrated neurophotonic probes come in. Here's a picture of an optical fiber coming onto a chip, coupled through a thing called a grating coupler, a little like diffraction grating. It takes the light and then it propagates it horizontally on the chip in this little, kind of like a little rectangular piece of material that guides, it's a waveguide for light, integrated waveguide. If we actually have different channels, different communication channels encoded on different colors of light, we can pick off those different colors of light and deliver the signals that are separately encoded on those different colors to different parts locally. This is called wavelength division demultiplexing. It's used in telecommunication to send many, communication channels down an optical fiber, and we're going to make use of it here. Okay? Yes? Can you speak up, please? Sorry. So I'm going to talk about that now. So thank you for the question. These constructs, these ion channels that can be stimulated by light, have some sort of wavelength range over which you can cause them to open up. Okay, that's their absorption band. And there are different types of, these are called opsons, this class of molecules that occurs in nature, and they all have things, absorption bands in the visible. What we're going to do is we're going to create narrow channels all within the absorption band of these opsons that we can deliver different signals at different places. So we're going to encode, we're going to channelize that absorption band. And so here is some of the first integrated neurophotonic probes that we built. Well, no, there were a whole bunch before this. These are the working ones. Again, the same sort of form factor, but if you actually look at them more carefully, we've replaced all of the electrical things with optical things. So here's the probes. What we're doing to actually separate the channels out and then to send to different waveguides down on the probes to a different little emission pixel at the end is a thing called an array waveguide grading. If you use an electron microscope and look at what we've created using nanofabrication technology, this is a whole bunch of separate integrated waveguides that acts like something like a diffraction grading. Interference at the beginning and the end allows you to separate the channels as a function of wavelength. And so now, if you actually look and you stimulate these things, you can see the local little points of light. No longer a big bolus of light going into the brain, but in fact, we can locally stimulate using these integrated neurophotonic probes. And this is all the heavy lifting of Aaron Segev, a postdoc in my group in physics. What's really cool is to actually put these things in brain tissue or in solution and look and see how strongly collimated the beams are coming off these photonic probes. If you actually look at one of the emitter pixels, you can actually see the first order beam. It's a diffraction grading that actually allows you to send a beam down the shank. You use a diffraction grading, and it couples it perpendicularly out. And you actually have higher order diffraction patterns coming off. The beam is very collimated. In fact, as you go hundreds of microns out, it still stays roughly the size of a cell body, a neural body, a soma. Okay, so this looks really good, and we've done first experiments. We've taken Aaron, took these probes to Andreas Tolias's group at Baylor College of Medicine in Houston, and spent a couple of weeks there, and along with Jake Reamer, they did experiment in mice that were transfected both with the effector and the reporter, GCamp6 and the opsons, channel rhodopsin. They could stimulate and record in the optical domain and proved that we can actually do single neuron excitation. Not a bolus, but now we actually have the tools to do patterned excitation.
Can neurons be stimulated from light outside of the visible spectrum, or does it just have to be?
We hope they can be, but the reporters don't exist. So I'm going around like a Pentecostal preacher with my evangelical mission to all the molecular biologists I can think of saying, we need infrared reporters. The reason why we want infrared reporters is because trillions of dollars have been spent on silicon integrated nanophotonics for telecommunications, and it's all in the infrared. So if I say, can you build me visible stuff? That's not what foundries do. So we've built all the visible stuff ourselves. There are some foundries, smaller foundries. In fact, Leti and Grenoble is doing some stuff in the visible. But the trillions of dollars have been spent on infrared, and we don't have reporters for that. So there's a huge reason why we, this mass production and delivery, we want to tap into the existing technology, not create new trillion-dollar costs, for developing the technology.
And so with the infrared, if we had the infrared light stimulation, would that open new discoveries?
Yes. Let me tell you like a crazy thing. This is like sometimes DARPA is the Defense Advanced Research Project Agency, and there's some really sort of futuristic, wacko, guys that are program managers there. You can imagine taking optogenetic constructs, if we had them for the infrared, transfecting the retina with that, and now people can do infrared vision without infrared goggles. That's crazy shit. Okay. All right, so I'm going to just conclude then with recording. So that was about stimulation. How do we actually do the recording? So let's think about the probes. Chris, how am I doing time-wise?
It is 10 past 11. And
Is that more than an hour I've gone?
no, it's coming up to an hour.
Okay, great. So to think about how we do the recording, this is not, I'm sorry, let me back up a minute. This light sheet microscopy and the raster scan stuff, the way you actually look and see which cells are lighting up because of calcium activity using optogenetic recording, you just image it using a microscope objective. Okay? You just look at the green light coming out. What we're talking about here is lensless system for collecting photons. It doesn't form an image. We're going to do something in the optical domain to begin with. Begin with means for the next 3 years. That is going to be lensless collection and triangulation. It's the optical analog of spike sorting, okay? So why is that? Well, I can think about breaking up that millimeter volume of Cortex into what I'll call unit volumes that have the granularity of the finest recording element in the finest pixel in our system, which is these detector pixels that have a dimension of about 50 microns. And so the unit volume has a thickness of about 50 microns, and it's established by the adjacent probes. Okay, so if I tilt the thing on the side, there's the 50 microns. It's about 282 microns, square root of 400 microns divided by the square root of 2. And we're going to have emitter pixels and detector pixels stimulating and collecting the emission. We stimulate in the blue, we collect in the green. Okay? And the granularity, again, is because of the size of our detector pixels. Now, what's interesting is that what we've done with this paradigm is we've put our detectors close enough together where they're more closely spaced than the optical attenuation length, so we can get all the photons. Okay? A little bit about the Avalanche photodiodes. This uses a full CMOS process. Our first chips for the detector pixels have come off the 130 nanometer node IBM processing line, one of these billion-dollar foundries, because you not only need the photodetector, but you also need a quenching circuit, some transistors on the probe itself. So when the photon comes in, it fires an avalanche. That's why it's called an avalanche of electrons. Basically, the little back bias diode breaks down, and then you have to have a quenching circuitry to turn off that cascade, and that happens in the picosecond time scale, and it sends an electrical impulse up the probes themselves. Ken, my buddy, is an expert in building these for about the past 10 years. He's perfected these for biological applications, and so he's done the heavy lifting in the technology of the unit devices, and so we're actually incorporating these onto our shanks now. So how does this paradigm actually work? Optical functional imaging using these probes, integrated neurophotonics. So looking at the top down at one of these unit volumes, this is a sparsely stained, using rainbow technology, slice of the cortex, and then I put in little cartoon elements, since we're building this stuff, so now you get cartoons and promises rather than real data. That's how science works. And these are 4 adjacent probes, so this is like a 282 micron on a side section, 50 microns or so. So on the edge, we have edge emitters. We shine blue light, a little 2 picosecond pulse of light. The chromophores of the GCAMP6 molecule fluoresce for about 5 nanoseconds. We collect those photons, not only on the proximal SPADs, single photon avalanche diodes, but as I said, there's something like about 20 that are within an optical attenuation link that will still be collecting photons. Okay? Yes?
Is there some way of detecting neuron external to the skull?
It's difficult because the skull, you can use, yes, but not with this kind of resolution. And if I could defer that question later, it would be wonderful like some of the kind of way out there guys in defense and DARPA and the Defense Advanced Research Project Agency say they want to have non-invasive imaging. They want to also do chemical sensing from miles in space and so on. I mean, there's some laws of physics that have to be respected, right? And so it's very difficult to get enough photons through the skull. Some get through, but very few, without scattering and so on and so forth, to actually do this kind of stuff. Okay, so I mentioned optical spike sorting. So first of all, let me tell you about the measurement space. What we're going to try to do is in this volume, this unit volume, which is set by the granularity of our detectors, there could be up to something like 500 neurons that are simultaneously active. How are we going to deconvolve all this simultaneous activity and spiking so we can actually do the analog of spike sorting with this 50-to-1 multiplicity for each detector site that I mentioned is necessary to avoid disturbing the brain tissue unduly? Well, the first is we better have a sufficiently rich measurement space, okay? So we have with 18 e-pixels within an optical attenuation link that can address that particular volume. Just turning them off and on gives us something like 250,000 combinations, illumination patterns. We have 18 detector pixels, so we can use those to independently from different position collect photons, and the product of those gives us something like 5 million different combinations. That's the richness of our measurement space. Now, these are not all independent, but a significant number of these things can effectively be made quasi-independent. Okay, so we have something like 400 to 700 neurons. Probably several hundred of those are active at any particular time. We have a complexity in our measurement space that greatly exceeds that, and so there's at least hope at the outset that we can actually do this deconvolution. Now, we being my graduate student in physics, Joe Redford. Why do I say my graduate student? A graduate student in my group, I don't own him, Joe Redford, and senior research scientist, physicist Laurent Moreau, who's an expert in photonics, are doing simulations as we're actually building the device, because we want to make sure after we spend a lot of money and a lot of time building these things that we're not fooling ourselves that this is going to work. And so this gives you a flavor for the simulations that they're doing. This is a single neuron. We know all the numbers about how many optical reporter molecules we can put in a neuron without disturbing it, how much intensity of light we can actually shine on the neuron without disturbing it, how many photons we're going to get out, what the quantum yield of the fluorophores are, and then how much, if you move your single photon avalanche diode away from where the neuron is emitting the photons, how many of those photons you're going to collect, and integration times, and so on. And so we have detectors on the front and the back of each of these shanks. These are meant to represent shanks, but in fact, these are just little rectangular boxes representing the single photon avalanche diodes. And I'm color-coding here, or Joe color-coded here, according to the rate at which these photons are actually going to be coming in from these fluorescing neurons at 515 nanometers in the green. Red means a lot of photons, fast rate. Blue means very few photons. So this is for one, we think of it like a point source, even though it's an extended neuron, about 15 microns in diameter, for photons. Okay? So if you think about all 12 shanks compactly represented in a rectangular grid where the horizontal axis is indexed according to what shank you're talking about, and the vertical axis shows the 74 detector pixels, single photon avalanche diodes per shank, front and back. You can see that the most proximal probes give us a fairly high photon count rate, something almost about a megahertz, 1 million photons, 1 million counts per second. These are working in Geiger mode, so we get a click every time a photon comes in, single photon detection. The good news is our dark count rate, there's always noise, right? Even when you have no photons, you get some clicks, right? And those come in at about 8 kilohertz. So many orders of magnitude separation between the signal that we're looking for and the noise of the system. And these are the guys that are working on this. David Gidney is actually the circuit designer in Ken's group at Columbia University, who designed the actual form factor for these single photon avalanche diodes. Joe is actually working at Caltech to make these into shanks that we can implant. So I'll just conclude by giving you a feeling for how this demixing is going to work. Here's our little volume of brain tissue. We think of basically some of these neurons, could be a large number of the neurons, depending on what tissue. Cerebellum, almost everything is active. The cortex, fewer things are active. So the activity rate generates these changes in optical susceptibility of these reporters that are in the molecules. We can tune the temporal excitation of these things in different illumination patterns. There's one illumination pattern, happens in 2 picoseconds. And then over about 5 nanoseconds, green light emerges. We can do this something like 1 million times for every action potential in the neuron, okay? Because there's a huge separation, nanoseconds for the fluorescence to milliseconds for the spiking. So we can explore in the same spike pattern, the same individual spike that a neuron is happening, we can try a whole bunch of different illumination patterns. And then from all of this data that we collect, we can do a very computationally intensive thing, which is basically demixing all of these photon counts to actually represent then the temporal records of activity of each of those neurons. And I have more stuff, but I left it out of the talk, where we've actually looked at this demixing process mathematically and proven to ourselves that this is a well-founded thing. What about the other brain fields? I'm getting to the conclusion of my talk now. Everything is possible with this photonic system if we actually have the reporters that sense chemistry, sense forces, sense electrical potentials, and change optical susceptibility. And so I mentioned this thing about being a Pentecostal preacher telling molecular biologists we need new types of reporters. Really, the sky is the limit if we actually have these particular molecular constructs to actually go in and be the front end of our measurement system. So I mentioned that I think we can get to 100,000 neurons for electrical recording, and that's kind of the gold standard. And then I mentioned this crazy new idea, I hope it's not so crazy, of integrated neurophotonics, and so far it's looking pretty good, but we still have to get first data using the system. But it's following on what's done in free space. We're just integrating it into these implanted, distributed lattice of emitter and detector pixels. So I talked about optogenetic stimulation. We've done that. Now we're building the detector pixels. We'll co-integrate that. And we promised in 3 years to actually have first data from this system. So we still have 2 years left. I think that over the course of the next 10 years, we can actually realize not just these optical spike sorting systems, but if we go to 2-photon fast optics on these chips, we can actually make imaging systems, range-finding systems that are lensless. This goes under the rubric of tychography. You can look it up. It starts P-T-Y, funny word, but we can actually make these and insert them in the brain and really do full imaging. That's going to take us at least 10 years to realize. First, sort of baby steps, not so baby steps. I mean, this is hard integration stuff. So summary and projections. Optical imaging is great because we can highly multiplex our measurements. But we can only go 1 millimeter deep with free space optics. And so that allows us complete coverage of really simple species like zebrafish, which are good models for zebrafish, not for humans. Right? When we want to go to the mouse brain, we can only access the top. You can't get on the bottom because otherwise you have to cut the neck off of the mouse and you're no longer actually measuring anything that's alive. So there'll be no spiking. So you can only get the top of the brain and only superficial coverage part of the cortex. And it's completely hopeless when we scale up to the human brain. So we need new paradigms to actually do this big science of trying to record from millions of neurons in active, freely moving and healthy species. So I talked about a new paradigm called integrated neurophotonics, where we can massively multiplex using wavelength division multiplexing and techniques from optics that allow us to put many channels on fibers, same sort of thing at the nanoscale. complex pattern stimulation that are more akin to the real kinds of stimulation that occurs in the brain. So this could be ideal for brain-machine interfaces that have very high finesse, tapping into all sorts of things that go on in our brains, ultimately. Multiphysical detection of brain fields, and not reinventing technology, but using silicon-based technology to realize it all. I'll end there. Thanks very much. It's been a pleasure talking to you guys.